Starting with the second part of today's symposium. The next speaker we have is Dr. Sivarajan T. He's like, it's difficult for me to introduce him because he's actually a dear friend of mine and a colleague also. So anyway, Dr. Sivarajan received his PhD from NCCS Pune, where he worked with neural and cancer stem cells. He's always associated with the big name in academia, like Indian Institute of Science, where he worked on host-parasite relationship with application of omics. Dr. Shiva worked with world-renowned John Hopkins Medical Center for over 3 years as postdoctoral scientist in the Department of Radiology and Radiation Oncology and Molecular Radiation. He was the lead scientist in developing the mini bioengineered liver tissue at Pandorum Technologies for drug screening, disease model, and bioartificial liver to support liver function in patients with impaired liver functions. Very recently, he has joined Archeon Group as a senior manager R&D and will be setting up a CRISPR and CAR-T facility for Archeon Group. So without any further delay, I welcome Dr. Shiva, and Dias is over you.
Thank you very much, Sachin, for the introduction, and good afternoon, everyone. Sachin, I'm just, for the first time, I want to share the slide. Let me check.
Yeah.
Okay. Can you guys see the slide?
It's loading. Yeah, we can see the slides.
Okay. So I wanted to attend all the sessions, but there were meetings. So fortunate to attend the previous one. Fantastic talk, something that brings up my memory because we were also doing similar stuff in Pandorum, and I can understand all the troubles that they went through to get the data. So my talk will be slightly different as compared to the other speakers. This will be more focusing on some basics, how 3D tissue engineering developed slowly, one by one. So let me get started. I would skip this slide because Sachin has already introduced me. So let's talk about organogenesis. Organoids are nothing but a mini organoid. Organoids are nothing but mini organs. So where do we learn from? From what do we learn? We learn from organogenesis. Organogenesis is nothing but when a egg gets fertilized to form embryo, the single cell divides to make its own copies, and then later on divides to form 3 basic lineages: endoderm, ectoderm, and mesoderm. And the organization, the 3D organization of these cells is what makes the different organs of our body. And this is so fantastic, and there is so little known about the whole process, is what we are trying to recapitulate now using modern techniques. There are certain issues that still remains to be answered. For example, what are the gene expression patterns when such an organ has to be developed? What causes the shape and size, the orientation, the roles of different signaling molecules, Wnt signaling, other pathways that influence the gradation of the organ formation. Does gravity have any effect on organogenesis? All this needs to be explored. When we do a PubMed search for keywords like organs, organoids, organoid, 3D cell culture, you can see a sharp increase in between 1965 and 1990s. This is sort of a false positive because the papers just picked up any work that was done on organs. So let's not consider them. But once you see from 1999 or 2000 onwards, you see a sharp increase in 3D cell culture. All this is because there has been certain scientific breakthroughs which allow people to study 3D structures. Let's say why we want to study 3D and what is the difference between 2D and 3D. So everyone who is right now doing culture work, most of them would have worked with 2D cultures. So 2D cultures are nothing but, you take single cell suspensions, put it on a plastic surface, and within hours they will attach and form a single layer, and then you're ready to do your assays. 3D is not like that. You have to design add the type of cells, bring it to a 3D structure. So the 3D tissue will only be ready or stabilized, and it will take days and sometimes weeks to get them ready. The quality of culture. So in 2D, you can just keep on redoing them. Trypsinize plate, it's very easy. It's very difficult to do 3D. But what are the caveats? What are the caveats of 2D? So in vitro imitation. So 2D does not imitate how it will look inside a body, but 3D would be very close to how it should be inside the body. So in 2D, there are very limited cell-to-cell interactions. It's just a layer of cells on a plastic surface. While in 3D, you have interaction between the cells in different dimensions, and also interaction of the cells with the extracellular matrix, which is a very, very important. That's what I'm going to talk about today. And then also cell morphology changes. You take a cardiac cell and put it on a petri dish. It just looks like thin layer of some beating cells. Its heart is not like that. Heart is 3D. And also when it considered 2D, you just give it ample excess amount of nutrients, oxygen, signaling molecules, which is not true for a 3D environment, where there will be a gradation, a variable oxygen supply, variable nutrients, which is very important for how the 3D tissue performs. So, well, why focus on 3D? So 3D better recapitulate in vivo cellular responses. Architecture of the 3D plays a very important role in diffusion of compounds. There will be cell-to-cell and cell-to-microenvironment factors. Again, in 3D, you can do co-culturing, and this co-culturing will be also having a fourth dimension of time. Which time the cells are coming in, what types of cells are coming in, will there be a migration, how angiogenesis happens. And also, when you're studying disease models, the pathophysiology is more similar to the in vitro system. So let me take you back in time where very small discoveries have led or paved ways to how we are doing 3D cultures today. So in 1906, about more than 100 years ago, Harrison wanted to study nerve fiber outgrowth. So how he did it is he took mouse embryonic nerve cord, and he put it in a drop of lymph on a cover slip, inverted it and fixed it on a cavity slide. And then he observed, and you give a three-dimensional space, these nerve fibers came out of the tissue. Then in 1926, tube cultures were invented, but they were little difficult to work with. So in 1929, Fell and Robinson made an ingenious method where they took watch glass, added a plasma clot, and on top of it, they were growing dissected tissues. And this was all put in a chamber of petri dish having some damp woolen cloth to prevent evaporation. Then in 1952, Moscona and Moscona did a very interesting experiment. They took cheap chick embryo organoids, and then they disturbed it by dissociating them, made it into clusters of cells. And then they let it sit, and within some hours, they saw the whole organoid came back together, basically telling that in development, even when you disturb the organoids, they have self-assembly and self-organization abilities. Then in 1954, tissue section cultures were invented, where in a media you will put a grade of metal, and then on top of it you will place a lens paper, and you put sections. You section the tissues and you put on top of it and do studies on them. Then in 1956, a very crucial invention was done. So Herram and Gay, they were able to isolate collagen from rat tail tendons, and for their surprise, the collagen that they isolated was transparent. This made the whole community of animal cell culture very happy, because then it becomes very easy to do microscopy on them. You can see the cells through the transparent gel. And another very important discovery was by Lasfergis, who discovered or isolated collagenase. So he isolated it from a bacteria called Clostridium hemolytica, and then using that enzyme, he was able to dissociate mammary glands and made mammary organoids out of it. So the next 10 years, people were having a hands-on to the collagen. So they were trying to do a lot of fine tuning. And from 1960s to 1977, they were trying to see what stiffness of gel is best for growing the cells, what concentrations. And then in 1973, fibronectin was discovered by Gramberg. Now, previously it was thought that ECM is just a gel forming solution, but the interaction between collagen, N-actins, fibronectins, lamellins, they play a very crucial role in cellular morphogenesis and cellular proliferation. So using these gels in 1975 and 1977, people were able to do floating collagen gels, which was basically sort of pseudo scaffolds, and the cells actually grew very well on them, and they also showed polarity. And in 1977, Orkin was able to isolate a gel from chondrosarcomas of mouse. They called it as EHS, which is 𝐊𝐨𝐬𝐻𝐭𝐞𝐫𝐧𝐚 Swan gel, which is also known as matrigel. The beauty of this gel is you keep it at low temperature, it will be liquid. When you bring up the temperature, they will be solid. So it becomes a carvable gel. So you can make these different shapes and designs out of it. Then in 1979, from the matrigel, people were able to isolate lamellin, which also plays very important role and is an important component of ECM. In 1984-85, certain experiments by Lee et al. Showed that in floating collagen gels, when you put memory epithelial cells, they show polarity, and because of the polarity and differentiation, these memory cells also excreted casein or milk proteins into the media. A fantastic experiment. And then in 1991, it was finally proven that ECM components like lamellin, fibronectin, collagen, they interact with integrins, which later on, which are part of the cells which activate certain genes responsible for morphogenesis of the cells. In 2001, using 3D collagen cultures, people showed memory glands branching out, a very important developmental study. And in 2006, Nelson et al. Were able to find out that TGF-beta is an anti-morphogenic protein. So if you have a gel, 3D gel, where you micro-pattern the cells and you give gradients of TGF-beta, the cells will migrate in a gradient fashion. And in 2008, Iraku et al. Were able to make brain organoids from embryonic stem cells in 3D structures. And in 2009, Sato et al. Identified a very important marker called NGR5 positive cells. These are actually native progenitor cells which are found in many organs of the body. And when you isolate the LGR positive cells, they themselves form organoids because they have all the pluripotent properties. In 2012, Nakano et al., they were able to form 3D eye cups from embryonic stem cells. And in 2030, Lancaster et al. From iPSC derived stem cells were able to form micro brain tissues or brain organoids. Now we talk about all the terminologies. Let me get it very clear, the difference between spheroids and organoids. The spheroids are simple clusters of cells. They are basically formed when you put the cells in a non-attaching. So they have nothing else to attach but to attach to themselves. So they all stick together to form a spheroid. And they don't require any scaffold, and they are just simple circular ball of cells. Well, organoids are complex. They are a cluster of organ-specific cells such as those from stomach, liver, and bladder. One of the key criteria is organoids should be having some sort of a stem cell in them, which are characteristic of that particular tissue. And the properties that they have is self-assembly, and a little bit of scaffolding or extracellular matrix is needed when you want to make organoids. Let's see some of the differences between spheroids and organoids. So no scaffold required for spheroids. Organoids will need some scaffold. Stem cells are not required, or you can actually have spheroids having the stem cell, but it's not mandatory. But for organoids, you need stem cells. Spheroids will be difficult to passage, while organoids can be passaged. Spheroids will have hypoxia present in the center, where organoids will not have. Necrosis develops with time because there is no stem cell to regenerate. Necrosis will be minimal in organoids. they will be only spherical shape, while organoids will have variety of shapes depending on the type of organ that you're working with. And for spheroids, you need a non-adherent culture condition, and for organoids, you need an adherent to ECM or scaffolds. Now, what are the types of 3D cultures that are that you can work with? So one is matrix embedded, where you embed the cells within the matrix and make shapes out of it. The other method is matrix assisted, where you make a matrix scaffold, and then the cells will come and attach to them. And the third is matrix free, where you form spheroids basically. Just to go into more depths, matrix embedded is you take the matrix, dissolve the cell or suspend the cells in the matrix and then print. Printing could be extrusion based, inkjet based, or laser assisted bioprinting, and then you have the different shapes and structure. Matrix assisted is you make the shape and then you ask the cells to come and bind to it. And then matrix free is very common practice that happens everywhere, is you do a hanging drop. So you have cells in a hanging drop of media, and then they come together and they form a sphere. Or nowadays you can even use ultra low attachment plastic, where the cells will come together because there is nowhere else they can attach to. So there are some of the, just to give an idea, there are some leading companies in this field like Organova, Insphere, Scifuse, N3D, and Tivido. They are the leading in actually bringing out a product into the market along with Pandorum technologies. And there are certain technologies that I would like to focus on. For example, the hanging drop method, if you want to do it in the lab, it will be very, very difficult to do. Imagine making the hanging drops and then feeding them every day. So Inspiro came out with an ingenious method where they use, patented technology, where on the top of the plate you have a small orifice, and you can make a hanging drop out of it, and media feeding and replenishing can happen from the top. But this is just a temporary way. Once the spheroids are formed, they have made another method where they call it as gravity trap. The spheroid falls down into the trap, and these grooves actually help in automating the media replenishment. When we talk about organoids, the final aim is to make the organ. So this is just a slice of liver to show the complexity of an organ. So you can see here, this is the central vein, and these are the portal artery and portal vein. So blood that comes from the intestine, which is full of nutrients, will come to the liver, and from the liver it gets filtered to the portal vein. Now this process is very important because liver is a homeostasis organ. So it decides how much nutrients is coming in, how much sugar is coming in, what to do with them. So the liver cells play a very important role in homeostasis. Imagine how we will build this. So there are certain technologies which I would want to talk about in brief. So one company called Scifuse used an ingenious method. So what they did is they made spheroids out of hepatocytes, and then to give it a shape, they used a very old technique called Kenzan. Kenzan is basically you have a base which have needles, and on top of the needles, people were putting in flowers. So this is sort of making a buffet. So then the needles, the flower will sit. So they modified the same technology where they had a robot pick up the spheroids and put it on the needles to form different shapes. It's like skewers, or in India we call it as seekh kebabs. So like that, they put all the spheroids together, let the spheroids fuse, and once they fuse, they take it out of the needle to give you this final tissue. It's an ingenious method of making units of the tissue to the whole organ. So now, why do we want to study 3D? As the previous speaker also said, when you want to study drug toxicity, what is better, a 2D system or a 3D? And definitely a 3D system will give you a more in vivo condition results. Disease models, again, when you talk about parasites, for example, malaria parasite, or even today the coronavirus, you need a system to study them. How do you do it? If you have a model system like a model lung or a model liver, they will become media to study the diseases. also you can use the organoids to create extracorporeal support systems like a liver dialysis system, which can be present outside the body of the patient, and blood can be circulated and it will function as a liver. Due to a confidentiality agreement with Pandorum, I cannot show all the data that I did in Pandorum, but there are some data that I can share which is publicly available. So you here on the left side, you can see Pandorum liver spheroid, HND cross section of that, and uh, this is the mesenteric staining where you can see the islands of hepatocytes within the larger tissue, and some staining of liver specific markers which otherwise in 2D you will not see, like the CYP3A4, albumin, bile secretory excretory protein, which form channels within the tissue. And one of the limitations which even the previous speaker was talking about is imaging the 3D tissues. 2D tissues are very thin. It's very easy for the confocal imaging to do, but when you come to 3D, the thickness itself will not allow